Every 15 seconds there is a child dying from malaria in the world. We here in the James lab are trying to make transgenic mosquitoes that cannot carry the malaria parasites. My name is Ole Torenius. I'm here at University of California, Irvine at the Department of Molecular Biology and Biochemistry in the lab of Anthony James. Today I will show you how to make transgenic Anopheles stephensi. We are doing this to make malaria mosquitoes refractory so they cannot carry the malaria parasite. Now I have prepared a vial with isotonic buffer where the mosquitoes are going to lay their eggs. I'm going to use this aspirator to suck up the mosquitoes and put in a vial. This vial is then going into an incubator that is 30 degrees Celsius, and we'll leave it for about 1 hour in a cup. Now we're going to pull the microinjection needles. These are needles made of quartz, and we have to use a very fine tip. Therefore we need this needle puller. We use a needle puller from Sutter Instruments and also the needles from Sutter Instruments. So now I've taken out the mosquitoes, and I will put these eggs down into the petri dish where I have a filter paper soaked in isotonic buffer. That is just to keep the embryos wet during the injection period. The amount of water on the filter paper is critical. You don't want the eggs to float around, and you don't want them to get dry. Now I'm going to move the embryos from the egg paper onto another 3 mm Whatman paper that has been soaked in isotonic buffer. And I do this to take away the chorion before aligning them up for the microinjection. So now I'm going to take away the chorion, which is this silvery surface on the